our Western blotting workshop. And yeah, I'm very happy to briefly introduce myself. So I'm sales director at Lucerna-Chem for those who were not in the part one. And we are the Swiss distributor of Agrisera since 2021 and enjoying a very nice collaboration together. And yes, we're using this opportunity to have an absolute expert in Western blotting with Joanna, and I will be very happy to hand over to her to start the presentation about the second part of this seminar. Thank you for joining.
Good morning, everyone. And I know that there are people from many countries who signed up for today's part 2 of the Western blot series. My name is Joanna Porankiewicz-Asplund, and I am a technical manager at Agrisera for the last over 25 years. I have been working with Western blots and antibodies. And I would like to share with you some insights which I have learned during troubleshooting hundreds of blots. And this presentation is recorded. You will receive PDF with links and also a link to recording. For those of you who did not participate in part 1, which took place last week. If you have any questions during my presentation, please type them in the chat box and I will address them later. And also, of course, there will be a quiz and you will have a possibility to win a free antibody of your choice. This is going to be at the end of my presentation. So part 2 is happening today, and would like to remind you about the part 3, which will happen on 27th of November. And of course, each session is going to be completed with questions and answers. For those of you who do not know Agrisera, I would like to show you where we are located. We are located in Sweden, in Scandinavia, and not at the polar circle yet. The company is celebrating 40 years anniversary this year, and we have 2 locations. Our antibody farm, located outside city of Umeå in this beautiful landscape. And in Umeå, we have a facility where we are doing antibody purification, quality control, and of course, we are shipping our products all over the world. In 2020, we became part of Olink Proteomics, and we are a production unit for Olink Proteomics platforms. And since last year, we are also part of a Thermo Fisher family. Our company is ISO certified. And we support you with primary antibodies for plant and algal research. If you have any ideas about antibodies you would like to have, you are always welcome to contact us. We also have quite a vast collection of epitope tag antibodies, which are validated on plant samples, few hundred of high titer secondary antibodies and detection reagents. So this is what we can support your research with. And workshop agenda. Last week, we talked about first part of the Western blot protocol, starting with protein extraction, and we reached the stage of the transfer. Today, I'm going to cover the rest of the protocol. So we have a ready membrane after the transfer, which can be done as in the option of wet transfer or semi-dry. And then I will talk from there until the detection and how a specific protein can be visualized in a complex protein mix using Western blotting. Just as a reminder from last week, repetition is good, how antibodies are making it possible to visualize the target protein. We are talking about an epitope here. So we are having an antibody, which is Y-shaped molecule. We have a constant region, so-called handle, which is not changing. And this is how we can purify antibodies to total immunoglobulin fraction using that part, which is binding to protein A or protein G. And then we have part which is very important for recognition of a specific protein. It is so-called antigen binding domain. This is a variable region where antibodies are binding to proteins or other compounds. And as you see here, we have linear epitopes, so subsequent amino acids in protein sequence, which can be from 2 to 15 amino acids antibody is going to bind. And this is the epitopes antibodies are binding in techniques like Western blot immunohistochemistry. So techniques where we are dealing with the natured proteins, but we also have antibodies binding to discontinuous epitopes. And these are brought by protein fold. So as you see here, two distant amino acids can be brought together by the protein fold. And these epitopes are recognized by antibodies in techniques which require recognition of a native protein like cheap immunoprecipitation. So please keep this in mind. This is very important for your future work with the Western blot and other techniques. So as you see here, we have, I prepared also links in this presentation, which you will receive. So if you're interested to find out more, you can read about it later. And now we're diving into the second part of the Western blot protocol, into membrane blocking, which is very important to make it impossible for antibody to bind to other proteins, preferably only to our target. And here I would like to point out we used tweezers not with flat ends, and please do not do like that in your lab. You can damage the membrane. But blocking can be done, as you know, in 4 degrees, or it can be done at room temperature for 1 hour. And as you see here in this, we are writing also that both membranes, PVDF and nitrocellulose, can be dried after blocking and used 1 to 2 weeks later. Why would we dry the membranes? The drying actually helps to fix the membrane and to fix the proteins on the membrane, to unfold them further, to make it accessible for the antibodies to bind, so to unfold the protein further. And in our lab, we are actually using this method. We prepare quite many membranes in advance with, of course, known sample setups, and then we store them for up to 6 months. First, we dry them, protein side up, and then we store them at room temperature, and then we use them at the later date. And actually, if you have sensitive samples where proteins can degrade in a freezer, when stored in a freezer, they will be safer on the membrane. If you want to share your samples with the other lab, you could also do it by sending them a prepared membrane instead of samples. We have tested this method for quite many years, and it's working very well for us. Membrane blocking reagents, I have ordered them from those which I found the most efficient with plant proteins. These are 5 to 10, this is 5 to 10% non-fat milk. This is a mixture of quite many proteins, which many times is more potent than BSA alone. However, there are always exceptions. We have also commercial brokers, which are often very efficient. For those who do not want to use protein blockers, we can use PVP40, which is a non-protein blocker. It's a chemical used in fractionation also, so probably available in your lab. BSA is down the list because what I have seen based on the result which we are receiving, oftentimes there is much more background if BSA is used alone. And at the lowest position on the list is gelatin. And here you can see an example of an experiment where the blocking was conducted for too short time, and the blocker was not potent enough. And the lab returned this result and said, well, antibody is non-specific, it's not working. I asked them to extend the blocking time, and the blocking was more complete. So as you see here, with such a simple adjustment, the outcome can be changed. And there's another situation when some antibodies may require blocking overnight. So this has to be tested experimentally. It's nothing what we can predict in advance. With antibodies which we are testing for the first time, we are actually using the protocol when the blocking is done 1 hour room temperature, but primary antibody incubation is performed overnight. And this is to allow the antibody to have the time for binding. But as you see here, the options can be different depending upon protein-antibody pair. So if you experience a lot of background, you may choose the option of blocking overnight. And how to remove unwanted background. This is also an issue which is often happening. And there is the possibility to do so-called adsorption. If we have samples with a mutant which is lacking your target protein, so there is no band here, you can incubate your primary antibody in a solution, of course, with a membrane where you have samples from the mutant. And what happens is that the unspecific antibodies will bind to the other proteins, and then you use this solution later after this adsorption, and hopefully the background signal will be decreased. It can be very troublesome to get rid of the background, and here is just a short description of a protocol, how to do it. You will also find it on our website. One can also try to get rid of Rubisco cross reactions using the membrane with Rubisco protein on it and do this adsorption, and follow with the same solution containing remaining primary antibodies to follow with this solution to the Western blot procedure on your desired set of samples. So unwanted background can be removed, but it is not guaranteed because if you have too little of, for example, Rubisco protein, which you want to remove the antibodies to Rubisco, if it's too little of Rubisco protein on the membrane where you're incubating your primary antibody solution, then it's, of course, not all anti-Rubisco antibodies will bind, and they can still follow and give you a signal when working with your specific primary antibody. But this method is definitely worth testing if you are troubled by the background. And I have also received a question, why there is small background when stripping the membrane. And it can actually happen, maybe some of you are doing it, that you are using harsh conditions to remove the antibodies from the proteins which are bound on the membrane. Well, when you are doing that, you actually also remove the target. So you may also remove the protein, your protein you want to detect from the membrane. And therefore, signal to noise ratio will be changed. And therefore, if you are stripping, then you may have increased unspecific background afterwards because there is too little of specific primary antibody to bind to your target protein. So you need to keep this in mind when you are doing stripping of your membranes. Of course, it can work well, but this method has also some limitations. And very often we're tempted to compare primary antibodies between each other. So, for example, the question was that a lab was working with not fully characterized antibody to protein of low expression, and then they got a signal from actin, but not from the desired target protein. And why is that? Well, I think that some of you maybe know what can be the reason. It's protein abundance here, which will make it that signal from actin will be visualized very quickly within maybe 30 seconds, while your desired target protein may require several minutes for visualization. So you cannot really compare antibodies with each other because they differ in also target proteins epitopes to which they are binding. And therefore, such comparisons should be avoided. And here a reminder for those who did not participate in part 1 that we have this wonderful protein abundance database where you can check abundance of your target protein before you start any Western blot experiments. This is crucial. I talked about it in part 1. If your protein is of low expression, below 10 ppm, you will require fractionation, high protein load per well for visualization of such protein. And also, there is, if some of you wonder if monoclonal antibodies are better than polyclonal antibodies, this blog post is answering that question. I can reveal that it depends upon the protein antibody pair, and we can never say that one type of antibody is better than the other. Loading control is important to use in your Western blots, and you probably recognize some of these proteins, or maybe you're using them as your controls. However, I hope that you have checked experimentally that these proteins do not change in the conditions of your treatments. Because, for example, tubulin, actin, they will change if your plants or organisms are stressed. One cannot choose a priori these proteins to be controls. It has to be experimentally proven that they are suitable for your setup. Of course, the purpose is to assess if we have equal protein load per well. And I really hope that you're loading based on protein and that you measure protein concentration in your samples, as loading per volume is not really appropriate. I will talk about this a little bit further in part 3 of this webinar. Antibody incubation buffer can be different. There are two options, TBST or PBST. And as you can see in this peculiar example, for this protein antibody pair using PBST buffer was actually producing less background. But as I mentioned before, it depends upon specific protein antibody pair. And PBS buffer should not be used when working with phospho-specific antibodies. And I see the question. Okay. What about loading depending on the chlorophyll? Yeah, chlorophyll is fine. Chlorophyll is fine. There is a certain proportion between chlorophyll and proteins. For photosynthetic organisms, I would use loading. I've done it, loading based on chlorophyll. And for other tissues, which are not photosynthetic, then loading per protein. And there are also reagents which will allow you to measure protein concentration in presence of denaturants. So there is a possibility to actually measure protein concentration in presence of SDS, urea, and other compounds. And now we are coming to the excitement of incubation. We are going to visualize our target protein, and I would like to show you how you can save time when you're doing primary antibody incubations. So in this example, yes, you see correctly, there are 3 antibodies which were incubated with the same membrane at the same time, 3 primary antibodies. Of course, these antibodies were characterized before, So researcher knew which dilutions to use. And this, because sometimes we have very little of our samples, if the distance between the proteins which we aim to detect is more than 20, 30 kilodaltons, then we can apply this approach. But keep in mind that you need to know those primary antibodies to be able to mix them together and save time by parallel incubation. There is another method to save time, a second method to save time when doing the incubation of primary antibodies. Standard protocols take a couple of hours, but it is possible to incubate everything together at the same time. Blocker, primary antibody, and the secondary antibody incubated at the same time, simultaneously. And that way, Western blot protocol is shortened to 1 hour. And of course, this is recommended for protein of high expression and antibody protein pairs, which you know how they are behaving. This is an example of antibody to highly abundant protein of photosystem 2 PSBA, which was tested that way. So simultaneous incubation of blocker, secondary and primary antibody at the same time can help to speed up the protocol. And there is a third option. In this case, primary antibody is conjugated, and it can be conjugated to an enzyme or to fluorescent dye. And that way, the protocol time will shorten. However, you need to keep in mind that this method is not suitable for proteins of low expression, because these proteins will require enhancement given by the binding of secondary antibody, because there will be more secondary antibody molecules binding to the primary antibody. Secondary antibodies can come from different hosts, and they look like primary antibodies. They are Y-shaped molecules, and they have the antigen binding domain where they are recognizing your primary. And there is a whole collection of secondary antibodies can be done in different hosts. So depending in which host your primary antibody is produced, then the secondary antibody need to match it. And do secondary antibodies a different price? Yes, they do. So, but I will tell you about it in a moment. So we have a primary antibody binding to the target protein on the membrane, and the host was rabbit. Then we need to use a secondary antibody, anti-rabbit. And as you see here, there is a tag, which can be, for example, an enzyme or fluorophore. And by doing this kind of sandwich, the signal is going to be enhanced. And there is actually a difference how secondary antibodies can perform. You should look for secondary antibodies of high titer. And what does it mean? You can use them in high dilution. So for example, 1 to 10,000, 1 to 25,000 times. And this will minimize the potential background. Never incubate secondary antibodies longer than 1 hour at room temperature. There was once a lab which used a protocol which lasted 3 days in total. And of course, it resulted in the membrane was black. It was too much of everything. So never incubate secondary antibodies more than 1 hour at room temperature. It's not necessary. And if you want to buy a secondary antibody, check in what dilution you can use it. Because usually we concentrate on amount. We have 1 milligram of a secondary antibody, usually 1 milligram per tube. However, 1 milligram of the antibody, which can be used in 1 to 25,000, will last whole your thesis compared to the less potent antibody. So pay attention not to amount, but to the dilution you can use the secondary antibody at. And here, an example. We have a high titer secondary antibody, goat anti-rabbit IgG, horseradish peroxidase conjugated. Which one you would use for Western blot? The second antibody is adsorbed. It means that during purification process, it has been passed to columns with IgGs coming from different species. In this case, it's bovine, human, goat, mouse, and rat IgG. So if you work with plant proteins, which antibody would you use? The one, the simpler version or the adsorbed one? You can type your answers in the chat if you want. Okay, no answers coming. Of course, for the simple Western blot experiment, this antibody is going to do well. The adsorbed versions, like this one, may be useful for ELISA when we are working with the material coming from bovine, human, goat. If we work with plant proteins, we want to use adsorbed antibodies in immunohistochemistry, because such secondary antibodies will may have less cross-reactivity. But for the single Western blot, this antibody will be suitable. And now we are coming to the exciting target visualization. This example is showing you enzymatic visualization of a target protein, which is done if a secondary antibody is conjugated to an enzyme. And we have other also visualization methods. Secondary antibodies can be tagged with a fluorescent dyes, or the third method, when secondary antibodies are conjugated to also an enzyme, but this is a chromogenic product, so it will not emit light. So we have three methods of visualization of a target protein. We have chemiluminescent, fluorescent, and chromogenic. And what are the advantages of these methods? We need a CCD camera, of course, if we work with chemiluminescence. And background can actually still occur when using chemiluminescence, as you can see here. The mistake which was done here was that detection of the very abundant protein was attempted with a very sensitive detection reagent. So it has extreme low femtogram detection. And of course, there will be a lot of other bands showing up. So you need to keep in mind that if you work with proteins of high or moderate abundance, then the most sensitive detection reagent might not be a best choice, because then it is simply too much for visualization. And there are different advantages of chemiluminescent detection. We have sensitivity, of course. However, recording of a signal has to be done immediately. It will not last. It is appropriate for quantification and analysis of proteins present in low levels. But that signal will fade. So there is also a disadvantage in that. Even though when I troubleshoot Western blots, chemiluminescence is still the most commonly used detection methods. It comes, of course, with some flaws. As you see here, there can be issues, and there can be different reasons for it. Here is a checklist for you for the future. when you work with chemiluminescent detection and you experience a high background which looks similar to what I'm showing here, there can be, I would like to point your attention here, interference with plastic used to wrap the blot can actually also cause this background signal to appear. And maybe some of you have been experiencing so-called ghost bands, and the non-specific bands, there can be different reasons for them. The most common is this one: no target protein on the membrane, and therefore everything else is coming up. Because keep in mind, Western blot is about signal to noise ratio. If your target protein is it enough of it on your membrane, it is going to be visualized. If it's not enough of it, of course, everything else will come up. And here the ghost bands, which maybe some of you have seen when you were working with your blots, and the reason for it is too much of everything. So too much of a protein combined with too high concentration of primary and secondary antibody, and possibly also antibody mismatch if we use the wrong antibody, secondary antibody, for example. So you can get rid of ghost bands by analyzing your protocol and decreasing the dilutions of your antibodies. And I received a question, if something went wrong with the chemiluminescent detection, can I still save that blot? And can I redo it without stripping? And the answer is yes, you can. You just, if you didn't manage to record the signal, or something else happened, you ran out of reagent, you can still wash that blot and apply the reagents again and record the result if it's done within the same day. Now we're coming to fluorescent detection, which is second most commonly used in the blots which I'm analyzing. And as you see here, it allows multiplexing because we have two different proteins which can be visualized with different labels with which will emit different light and therefore different colors. So secondary antibodies are conjugated to fluorescent dyes, and this method is very useful for multiplexing, and also the signal will not fade. So this is a big advantage of using fluorescent detection. However, of course, you need CCD camera, and sometimes it can be lower sensitivity compared to chemiluminescence. If you have a luxury to have a possibility to detect using chemi and fluorescence, you can simply choose which method will be more suitable for your protein antibody pair. I have seen examples when by some reason antibodies provided with the CCD camera for fluorescent detection when were not matching the protein which was to be detected. So there can be, of course, different issues here, but it is a very useful method for visualization of also several proteins at the same time and their quantification. And Agrisera is also providing secondary antibodies conjugated with fluorescent dyes with so-called dye lights. And then the third method is colorimetric detection. Here we don't need a CCD camera. Secondary antibody is conjugated to an enzyme, alkaline phosphatase, and we are also providing the reagents for colorimetric detection. You can simply save the blot in your lab book, although it will fade a little bit with time, so it's good to document it anyhow. That method is cheapest, quick. However, it has low sensitivity. I would not recommend to use colorimetric detection, for example, for detection of transcription factors. It is prone to failure due to lower sensitivity of that method. And hopefully, we are enjoying our final results, which can be obtained in those using those 3 different detection methods. So we have chemiluminescence, fluorescence, and colorimetric detection. However, to be certain that correct target protein is visualized, you first of all need proper markers. And we are mixing these colored markers with IgG binding site. In this case, this is magic marks. And this allows visualization of molecular weight during development of your blot. This is very important. I've been troubleshooting results where markers were wrongly assigned and improper conclusions have been drawn because of that. So keep this in mind that these labeled markers are a very good way to get visualization of the molecular weight directly when you are developing your blot. I'm going to talk about this in a third part, which is focused on interpretation of Western blot results, because molecular weight of your target protein calculated from a sequence may actually be different from the apparent molecular weight which you observe on the blot. And there are different reasons for that. One of the questions was for tag antibodies, which were not giving any signal. And I was asked for reasons for this. And there is a whole list what can go wrong and what to do. First of all, if expression level of your tagged protein is too low, the antibody may not visualize it. Increase protein load per well. Sometimes use of harsh denaturants like this 6 molar RIA will help to unfold the protein and make it accessible for antibody to bind. You need to keep in mind that adding a tag may also change how protein is folded. And also Western blot, even though it's done in denatured conditions, actually many proteins may partially refold during that procedure. So there are different things to do. And also, if one tag antibody is not working, then the other one actually may do the job, depending which epitopes these antibodies are recognizing and which epitopes are accessible on your target protein. So everything is about the binding between antibody and the target. Agrisera is providing quite many of antibodies to epitope tags. Of course, the popular ones like GFP, but we also offer, for example, Turbo ID for protein interaction work, and quite many other quite unusual tags. So if you are searching for an epitope tag antibody, you're welcome to check. And one of the questions was how to get an intense signal from Western blot using a weak antibody. And of course, you need to go, instead of running subsequent Western blots, I suggest that you go to the desk, take a pen and start to analyze your protocol. Did you use correct sample type? Check where the target protein is mostly expressed. Maybe you should use 2 millimeters of a root tip, not a whole root. Maybe it is the senescent leaf. So correct choice of material for analysis is crucial. For proteins of low expression, fractionation is necessary. Then we have protein load per well. Maybe we need to increase it. And transfer conditions, are they optimal? For small low molecular weight proteins, they are often blasted through the membrane because we use the same protocol for all proteins irrespective of their molecular weight. So if a protein is to be detected is of 10 kilodaltons and you apply transfer conditions like for a protein which has 100 kilodaltons, you will probably blast it through the membrane. So the protein will not be there, therefore no signal with the antibody. It's good to use primary antibodies which are purified and appropriate detection reagents. So for proteins of low expression, we use most sensitive detection reagents, but of course we have to count that also the background signal may increase in such case. So all this is called optimization. This is crucial for your success, and it's not always antibody which is to be blamed. Here I would like to share with you some data. There's an example of an experiment where we have the outcome being several bands, and of course we see also a target protein, but these are the background bands. As you see, protein load per well was quite high here. Incubation was done overnight. In the optimization attempt, the protocol was optimized this way. And as you can see here, the only thing which was done was decrease of the protein load per well. This antibody is quite strong, and I think that incubation overnight is not necessary. You need to keep in mind also that if you incubate overnight, you may actually increase the risk for the background. So for some antibodies, it's better to incubate 1 hour room temperature or decrease protein load per well, as you can see in this example. Advice for beginners: record the correct band first before multiplying variations of conditions. This is the most common mistake which is done, that we are doing blot after blot after blot. What should have been done is the thorough analysis of your protocol, starting with a checklist of biochemical properties of the protein you are aiming to detect: molecular weight, isoelectric point, expression level in different tissues. This is crucial for your success, not only the antibody. And here, this is a properly validated antibody when the signal in the deletion knockout mutant is gone. And I'm going to talk about this in a third part of the webinar. Here I share our protocol, which we're using with antibodies which have not been characterized at all. We are using, as you see here, high protein load per well. We are blocking short, but we are using prolonged incubation in a high concentration of primary antibody, so 1 to 500 overnight. And of course, we don't incubate secondary antibodies longer than 1 hour. This protocol, in many cases, lead to increased background, but it will also show us if antibody is detecting target protein or not. So instead of applying different conditions, we apply too much of everything in a first shot, and then we take it from there, and it is easier to optimize such protocol and modify the conditions later. So for example, decrease protein load per well, use more diluted primary antibody, and so on. Advice for experienced Western blot user: look over the protocol that you're using, the same protocol that not fit all antibodies. And I hope that I was able to show you that it is applied protocol is very dependent upon protein antibody pair. So definitely the same protocol cannot be always applied. And if you have still problems with your blots, if they look like that, you're welcome to contact me. I often troubleshoot antibodies which are not coming from Agrisera, and I'm happy to share my knowledge. And if you send me the data, I can give you suggestions what can be done. And here I want to share with you some common misconceptions about the antibodies. So now I want you to answer for yourself. Other antibodies I have bought from Agrisera work for my setup. Therefore, this one should also work for the same with the same setup. True or false? Of course, false. The antibody works for species A, so for sure it's going to work for species B. True or false? Those who attended part 1, I hope you remember, this is false. Because one cannot assume anything, you always need to check that the sequence which was used to elicit given antibody is also found in a protein you aim to detect. This is most common mistake why antibodies are not working. The same protocol can be applied for all antibodies. That one is easy. This is false. This antibody gives a good signal in Western blot, so therefore it should also work in other techniques. What do you think? True or false? False, because each technique needs to be validated on its own. And you can read about the misconceptions if you are interested in those blog posts. So on our technical blog, we are sharing the technical tips, like the one I showed you with a different incubation buffer, and many other posts are coming regularly. Here's the link to our blog. And I would like to also point your attention into how we are citing the antibodies and why it's important. Please remember, because there is no worldwide standard, unfortunately, always include antibody name, product number, and supplier. Because this data is missing, more than half of antibodies used in research cannot be properly identified, unfortunately. Of course, it would be good to include also conditions of your Western blots, and I think you know why now. But this is what we are including on Agrisera product information sheets. This is for your help to know with which conditions given antibody is giving that result. So please help to increase reproducibility and include this minimum: antibody name, product number, and supplier. Companies may carry many antibodies. Agrisera has several antibodies to Rubisco, so it's not enough to write a protein name. You need to also provide product number and supplier. And now a little bit about free resources, which we are offering: educational posters focused on photosynthesis, epigenetic, protein extraction, and Western blot. I would like to encourage you to check Global Plant Science events calendar if you are planning your conference attendance next year. There are also a lot of online events which are free, which are included in that resource, which Agrisera is running together with American Society of Plant Biology for the 6th year. And we are also offering you free images for download, which you can use in your publications. They cover different schemes, model species, and proteins. And this year we have produced a Western blot poster, which is summarizing those webinars. As you see, it's a lot of QR codes which will lead you to specific landing pages. The poster is easy to transport. When it's folded, it's A4 format, and you can order it for free from our website. And this poster contains concise tips from those presentations in the graphical format. If you would like to hang it in a lab, you are most welcome. It's just to go on a website and request it. And we are also offering over 150 free antibodies in return for results. So if they are fitting your experimental experiments, your questions you are asking, please let me know. Please check the list. And now we're coming to the exciting part of sharing the link to the quiz. The quiz has questions based on the presentation, and you have a chance to win a free antibody of your choice. The quiz is going to be open until tomorrow morning, and then we will let you know who won a free antibody of your choice. I will paste the link and the QR code in the chat also, but you are also welcome to make a picture with your phone now. And with a picture from Agrisera surroundings, I would like to thank you for participation in part 2 of this webinar series, and I welcome your questions. So, are there any questions? You can also send the questions by email. So that's, of course, a possibility.
Joanna, there was a question in the chat.
Yeah.
When you said to dry the membrane, how do you do it? What temperature? Was
Yeah,
asked
room
by
temperature.
Anna.
You are air drying the membrane. So you take your membrane protein side up and you dry it in room temperature. It is described in a blog post exactly how to do it. If it's PVDF, it will dry quite quickly, and afterwards you can store it for up to half a year with a good result.
And there was another question by Harris. Why sometimes tagged protein disappears after a certain time?
Disappear. I think it may be connected with the fact that if you are reusing antibodies, then it may be that that antibody cannot be reused. And therefore, you may see that the target protein is with the subsequent incubation, the target protein is not showing up because most of the antibody bound in the first incubation. So the subsequent incubation, there will be no primary antibody to bind, and therefore there will be no signal. I talked about it in the first part of the webinar. Some antibodies can be reused many times, and some antibodies may be reused only once. I mean, cannot be reused. I hope that that answers your question.
Then we have another question by Odd. I was wondering, what is the difference in size between a primary antibody and a secondary antibody coupled to a fluorophore?
Oh yeah, I'm sorry I didn't answer that question during presentation. The reason the difference is not big. It is a couple of kilodaltons between the primary and secondary antibody coupled to fluorophore. Of course, there is a penetration difficulty in immunohistochemistry, and therefore, camelid antibodies will penetrate much faster because they are smaller, and also nanobodies, but it's always a limitation that we may not have this specific antibody type available for our target protein.
And maybe also to just tell also, I mean, in case you're looking for camelid antibodies, we do have some solutions also for custom production of such antibodies. So if you're looking for some of those antibodies, feel free to contact me. You have my email from the registration.
Yeah, you're welcome. Are there any more questions? You are always welcome also to send them to me directly. And if you have any results which you would like to share, you can also do it. Here's my email address. I'm always happy to help. So don't hesitate to contact me if you would like to have some help with your Western blots. And there are no stupid questions, this is for sure. I am taking each question very seriously because I have seen during the troubleshooting time, I have seen a lot of different strange things happening with Western blot technique. I've seen contradictory results. So nothing is strange for me if I say like that. I'm always surprised. So if there is no more questions, then maybe we should welcome you for the third part of the presentation, which is taking place on 27th of November. I hope that you found this material valuable. You will be able to access the slides and the presentation on Lucerna website, and Thomas will send you the links. And I hope that you also want to participate in the quiz. So thank you from me for today. I'm very happy that you wanted to join, and I hope that this is useful. And in case of problems, you're welcome to contact us.
Yes. Thanks a lot also from my side, and hoping to see you in 3 weeks in the next part. Thank you. Have a good day.